Bom dia a todos. Gostaria só de agradecer novamente, né, por essa experiência fantástica. Como a Solange disse, realmente tem sido de muito aprendizado para todos nós. Então, eu acho que daqui para frente a gente só tem a aprofundar cada vez mais, né, e intensificar essa aproximação, essa parceria. Então, eu agradeço muito a todos vocês e muito obrigado. Podemos iniciar.
Ok, antes de iniciar, pessoal, obrigada, Claudiozão, obrigada, Solange. Bom dia, pessoal. Bem-vindo todos ao segundo dia do módulo 2 do Programa ECHO. Nesse módulo, nós estamos abordando as questões de qualidade, produção e desenvolvimento com enfoque para a pesquisa clínica. Esse módulo ele tem sido liderado pela Heloísa, da Amgen, a Vanessa, da BMS e eu, que sou da Roche, para quem não me conhece. A gente tem apoiado a Heloísa, né, em todas as questões de planejamento e logística desse módulo. Só recordando para quem não participou do último, então, assim, esse módulo a gente começou no dia 9 de novembro, a gente iniciou com a Health Canada, né, em que a gente contou com a participação do Martin, da BMS, e do John, da Amgen, e foi bem interessante a apresentação foi gravada, depois vai ser disponibilizado para todos. E assim, um pouco antes da reunião, a reunião foi numa segunda-feira, e aí na sexta, a Anvisa endereçou alguns questionamentos para a gente, alguns pontos que a COPEC achava que seria interessante abordar na apresentação. Então, os nossos colegas, eles não conseguiram, não tiveram tempo hábil de incluir aquilo em detalhes na apresentação que a gente realizou no dia 9, mas posteriormente à reunião, eles responderam e a gente mandou todos os questionamentos da Anvisa respondidos por e-mail, mas assim, pessoal da COPEC, a gente segue à disposição, se não for suficiente, se vocês quiserem mais alguns esclarecimentos, por favor, só sinalizar para as meninas da Interpharma que a gente volta a entrar em contato com eles. Como a Solange já disse, hoje o nosso enfoque será FDA, e a gente vai contar com uma speaker da Roche de Genentech. A Cecília trabalha com a gente na Roche de Genentech desde abril desse ano. Então, assim, o nome da nossa speaker hoje, que vocês já viram a carinha dela, que ela já falou algumas coisas, o nome dela é Maria Cecilia Tami. Ela é atual líder da parte de política regulatória nos Estados Unidos, na parte de CMC, da Roche de Genentech. Ela é cientista, especialista em assuntos regulatórios, com mais de 25 anos de experiência regulatória no FDA, em qualidade de produtos biológicos e também em pesquisa básica em biologia molecular e virologia. Ela tem um Master Degree em biologia molecular. Ela é PhD em virologia molecular pela Universidade de Buenos Aires, na Argentina. E ela tem pós-doutorado na CIBER, do FDA, trabalhando com aspectos moleculares de picornavírus. E, em seguida, ela atuou no escritório de produtos de biotecnologia da CIDER, na FDA, e participou da avaliação de qualidade de produtos biológicos durante o desenvolvimento, licenciamento e pós-aprovação e nas inspeções de pré-licença e pré-aprovação de instalações para fabricação e para testes. Então, assim, hoje a gente está com uma pessoa assim, com bastante experiência com a FDA, vai ser bem legal para ela compartilhar com a gente. Gostaria de falar para vocês que essa palestra hoje ela vai ser dividida em 3 partes. E aí a gente vai ter pausas de 10 minutos para a sessão de perguntas. Só que hoje, como a gente está sempre atento aos feedbacks que todos estão caminhando, a gente vai tentar algo diferente hoje. Hoje o microfone de vocês vai estar liberado, então a gente pede para que vocês prestem atenção enquanto não estiver falando, para deixar o microfone no mudo, tá? Mas assim, hoje a gente vai deixar sim os microfones abertos para vocês poderem perguntar, mas a opção do chat continua. Então a gente tem 2 opções, ou por microfone ou pelo chat, e as perguntas podem ser feitas em inglês, em espanhol, porque a Cecília fala espanhol, e também em português. E com certeza a gente está aqui, a Elo, a Van, a gente está aqui para fazer a tradução do que for necessário. O importante é a gente aproveitar esse momento e interagir ao máximo. So now we are going to switch the language into English. Cecilia, I know that I had already said this before, but again, on behalf of Interpharma King Program, thank you very much for for being here with us today and share all your experience. And I'm now pleased to hand over to my colleague Maria Cecilia Tami. Thank you.
Oh, okay. Thank you. Hello. Good morning. Let me share my screen. Let me see if I guess this should be. You tell me whether I'm doing things correctly or not. And so now I think, should I change? Let me put the presentation, and then I think I have to check, then switch. Okay, let's see if I did it correctly. Can you see my screen?
We can see your screen.
Perfect. So, well, good morning to everybody. First of all, I want to also thank Interpharma for allowing me to participate today in the KIM program, and also everybody that is out there listening and taking part and participating in this training. I also thank you very much. So today I'm going to talk about the chemistry, manufacturing, and control aspects of an IND application, and mostly from the perspective of CDER, of biologics regulated at CDER, as I am a former FDA CMC reviewer from CDER. So we are going to walk together through this presentation, and I think I understood from Portuguese that Camila mentioned that we are going to do 2 stops during the presentation, where please feel free to ask questions that you might have. And then at the end of the presentation, we are going to have another Q&A session. So let's see. Well, this is a disclaimer, and also you can find here a contact information. If you want to contact me, you have any questions, please do not hesitate to go ahead, send an email, and I can answer anything that you may need. So this slide shows the outline of my presentation. I just prepare you psychologically. It's going to be long, but I hope that we can bear together and go through all of it and interact and discuss as we go ahead. So first I'm going to do an introduction to FDA with a brief outline of the legal and regulatory history of FDA. I will give you then an overview of the organization of FDA, of the centers that are responsible for regulating the drugs and biologics, and also of OPQ, that is the office that is responsible for product quality. I will then go and describe and give you some general information about INDs, why we need an IND, what are the kinds of INDs that exist, and then also share with you some infographics, some data on INDs that CDER receives. Then I will discuss and describe what is the process for the review of an original IND, and describe briefly the FDA expedited programs that FDA has to allow and facilitate the availability of drugs and biologics for serious diseases. We will go into the format and content of the IND, and then basically go deep into the CMC package. And finally, we are going to discuss what are the activities that are that the sponsors should do during the maintenance of the IND. Let me go through this one. So, as you may know, FDA does not only regulate drugs and biologics. FDA also regulates a series of other products like cosmetics, tobacco products, devices. And this slide just gives you a glance of the activities of FDA. Basically, there are about 400 licensed biologic products, over 20,000 prescription drugs approved. FDA oversees over 6,500 different medical devices categories, regulates about 77% of the US food supply, oversees 85,000 tobacco products. It has approved about 1,600 animal drug products, regulates, the FDA-regulated products account for 13% of U.S. imports and 19% of U.S. exports. And basically, FDA products are manufactured and handled at more than 270,000 facilities, from which half are overseas. So this slide shares some kind of milestones about the legal and regulatory history for FDA. So the first milestone was in 1902, where the Biologic Control Act was signed. This law was, the purpose was to regulate the commercial production of vaccines, serum, toxins, antitoxins, and similar products to ensure the safety, purity, and potency of these products. And this law was in reaction to the death of several children due to contaminated diphtheria toxin antiserum and smallpox vaccines with tetanus. After that, the next law that was the beginning of a series of laws to protect consumers was the Pure Food and Drug Act, that basically prohibited interstate commerce in misbranded and adulterated food, drinks, and drugs. And this was basically driven by the poor conditions in the meat processing establishment. And it was the first regulation for labeling. Basically, the label of the product had to have the active ingredient, so you need to know what was in the drug, and also some level of purities, the levels of purities. After the Pure Food and Drug Act, the Federal Food, Drug, and Cosmetic Act was enacted in 1938, that required that new drugs should be shown to be safe before going to the market, and this law gave FDA the authority to regulate food, drugs, and cosmetics, and it also was in response to death caused by ingestion of sulfanilamide. In 1944, the Public Health and Service Act was enacted, and this was the first regulation for biologics. In 1964, the Kiefer, I guess, Harris Drug Amendment was an amendment to ensure the drug efficacy and safety. So not only you should show safety, but also efficacy, and also disclose the side effects. And it was in response to the thalidomide disaster, that this was a sleeping pill that caused birth defects in a lot of babies. And finally, in 1963, the IND regulations were enacted in the Code of Federal Regulation in the Title 21, Section 312. So how is FDA organized? So FDA is a big organization. According to the data that I found for 2018, the number of employees is about 8,000 employees. FDA is an agency inside the Department of Health and Human Services, and is organized in around 9 centers and other offices. And so in the Center for Biologic Evaluation and Research and the Center for Drug Evaluation and Research are the centers that are responsible for regulated drugs and biologics. CDER, as I mentioned, is one of the centers that it's in FDA and oversees the drug program, which includes over-the-counter drugs. So those are the drugs that you can buy without a prescription, and also prescription drugs that include biologic therapeutics and generic drugs. CBER is the center in FDA that regulates biological products, which include blood products, vaccines, allergenics, gene therapies, and xenotransplantation. It also regulates human cells and tissue products, medical devices for the collection, processing, testing, screening, manufacturing, and administration of blood components, and human tissue and cellular products, and also regulates HIV tests for donor screen and to diagnose, treat, and monitor persons with HIV and AIDS. As I mentioned, I was part of CDER, so most of my talk is based on my experience in CDER, the principles of that I'm going to discuss with you today apply to other products, but that is why I want to show you the CIDR organization that basically has different offices, and the office inside CIDR that is responsible for overseeing the quality of products is the Office of Pharmaceutical Quality. The Office of Pharmaceutical Quality has around 1,350 employees, has different offices, and inside and oversees like all the aspects of product quality, including surveillance, policy, review, research, inspections. And I was part of, I was part of the Office of Biotechnology Products in OBP, and OBP has 4 divisions. I was sitting in Division 3. And the Office of Biotech Products regulates biologics, biotherapeutic proteins. There are biologics regulated in CIDR. As I mentioned before, the other biologics are regulated in CBER. And the Office of New Drug Products is the office that regulates the small molecule drugs. So let me go into some general information about INDs. So now you have an idea of the size of FDA, the granularity, the different offices and centers, and you can maybe have a sense of on the amount of people that work in overseeing the different aspects of submissions. So the first question is why would you need an IND application? And as I mentioned before, I talked about the Food, Drug, and Cosmetic Act, and this act, this law, requires that the sponsor have an approved application to transport the product across state lines. So requesting an IND gives an exception to this requirement, and basically it's a means to obtain FDA permission to ship an investigational drug or a biologic across state lines. An IND is needed to start a clinical study. All the IND regulations are located in the 21 CFR 312 that I also mentioned before, and the information in the IND should include information regarding clinical data that would support the drug is reasonably safe for initial testing in humans, manufacturing information, how was the drug manufactured, and then clinical protocols and investigator information, including commitments to obtain informed consent from the subjects that are participating in a clinical study, as well as having an institutional review board for your clinical study. There are different categories and types of INDs. INDs can be commercial INDs, and in this case, the sponsor is a corporate entity, or they can be research INDs, and the sponsor then is an individual investigator or an academic institution. So the types of IND that exist, one is the investigator IND. Basically, IND is submitted by a physician, that is the one that conducts the investigation with an approved drug, or also can be an investigation with a drug that is approved, that is in the market, but this investigator is testing in a different subject population for a different indication. The emergency use IND is an IND that allows the FDA to authorize the use of a drug, an experimental drug, in an emergency situation when there is no time to do the whole IND process. Exploratory INDs are INDs that involve a very limited number of subjects. They have no treatment or diagnostic intent. They are more a proof of concept IND. And then the treatment INDs that provide eligibility to subjects with experimental drugs that show promise in clinical testing for serious or immediate life-threatening conditions, while the final work is conducted in and FDA is reviewing the application. So basically, this IND allows subjects to continue receiving a drug outside a clinical study before or in the meantime when all the review process for approval of the drug is taking place at FDA, so that the subject does not stop its treatment while the approval process is ongoing. This slide shows you gives you a sense of the number of INDs, of new INDs that are received at CIDR over the years, from the year 2009 to 2019. The information is divided in three parts. In green, you can see the total number of INDs per year. In blue, those that are commercial INDs, so they are from commercial entities. And in red, the ones that are research investigator ones. These INDs do not include INDs for biosimilar products. But as you can see, the number of INDs that see their reviews per year is quite significant. So, how is the process for review of an IND, of an original IND application? The first thing that I want you to know is that the process for a revision of an original IND has to occur in 30 days. It's 30 days timeline. So from the receipt of the IND until the decision is made on whether the IND is allowed to proceed or is placed on hold, this is a 30-day time frame. So before the IND is submitted to the FDA, the sponsor can request a pre-IND meeting. This is not an obligatory meeting, but the sponsor can meet with the FDA, submit a series of questions, and get feedback from the FDA, and basically trying to understand the readiness for submission of an IND. These pre-IND meetings can be multidisciplinary, or they can be discipline specific. They can be, for example, CMC only, or they can have questions that are like for CMC, for clinical, for non-clinical. So as I said, these are not compulsory. So the sponsor at some point submits the IND, and then the review clock starts, and all the disciplines are going to review their piece, their part of the package of the IND submission. In the case of a CMC reviewer, the reviewer is going to review the information in the regarding CMC, that it will include information about the expression system, the cell banking, the drug substance, how it's manufactured, its characterization, release, stability testing, information about the drug product, formulation, manufacturing, release and stability, and also immunogenicity. The scope is for each reviewer, for each discipline, to determine whether there are safety issues. So when a reviewer looks at an IND, the assessment is based on identifying whether there are any safety issues in the package they are reviewing. If there are none, then the IND is allowed to proceed. But if any discipline found a review issue, then the sponsor is contacted through IRs. Depending on the timing, you can send an IR, or you can have to call the sponsor and discuss in a TCOM. Try to solve the deficiencies, ask for more data, or ask for rationale, and basically try to see whether there is additional information that could address that safety issue. If the issue is resolved within the 30 days time clock, then the IND could be allowed to proceed, and if it is not, it will be placed on hold. So, well, I mentioned already that the sponsor have the possibility to meet with the FDA before submission of their IND. And this is basically, again, to discuss the readiness of their IND application. The sponsor should request this meeting, and then 30 days before the meeting, they should submit a package. The package is going to have background information to some questions and background information for the FDA to have a sense and provide input. It can focus on specific questions for the review disciplines. There is only one pre-IND meeting per IND. And regarding CMC, basically discussions go around product qualities, safety issues related with identity, strength, quality, purity, or potency, and whether there are some identified potential hold issues that need to be addressed before submission of the IND. So when a sponsor submits an IND, the IND is received at FDA and sealed at FDA at the central document room. The first thing that happens is that the IND is assigned to a review division, to a clinical review division, and basically this is informed by the primary endpoint of the clinical study. in general. There are some other options like precedence, but basically an IND is assigned based on the review division, based on the primary endpoint of the clinical study. Once the review division is identified, then a regulatory project manager or RPM is assigned. This person is the point of contact within the review division and with the sponsor. This person is the one that communicates from FDA to the sponsor. There is no direct communication of reviewers with sponsor. Everything occurs through the RPM. The RPM issues an acknowledgement letter letting the sponsor know that the IND has been received. And then it's also the person that manages the IND review process, establishes the timelines, establishes the safety meeting, that is the internal meeting that all the disciplines have before the 30-day clock or due date or safety date to kind of come together and discuss whether there have been any safety concerns identified in the different disciplines. And then an assigned review team is assigned, and all disciplines are represented. So you have reviewers that are going to review the clinical information, non-clinical, other reviewers that review the non-clinical and toxicology information, others, like I used to do, that review the CMC, others that review the clinical pharmacology generally. There are two persons per discipline involved in an IND review, a primary reviewer and a team leader. And again, so this will make a group of around 10, 12 people that are reviewing an IND submission. So we mentioned the IND arrives, there is a review, a clinical division assigned, an RPM assigned, and a safety and a review team is also assigned. The review team reviews the IND information and determines within the 30 days of the IND receipt whether the IND is safe to proceed, so that the proposed clinical study can be conducted without unreasonable risk to subject or patients. Some divisions may send a safe to proceed letter to the sponsor, letting the sponsor know that the IND is safe to proceed and there are no hold issues identified. Or some divisions do not send a letter, but in the case the sponsor does not receive a letter within these 30 days of submission of the IND, then they can give for granted that the IND is allowed to proceed. So basically, no news is good news. However, if there are safety concerns that were identified by any of the disciplines and they couldn't be resolved within the 30 days of the IND review clock, then the IND may be placed on clinical hold. And this means that the proposed clinical study cannot start. In this case, the FDA must issue a clinical hold action prior to the 30 days. So if there is a hold, FDA has to actively let the sponsor know that the IND is not allowed to proceed. And an important concept is the fact that the FDA does not approve an IND. INDs are not approved. INDs are allowed to proceed or are placed on clinical hold. So what is a clinical hold? A clinical hold is an order issued by the FDA to the sponsor of an IND to delay a proposed clinical investigation or suspend an ongoing clinical investigation. What this means is that the FDA may prevent the initiation of a clinical study, for example, at the original for the phase 1 IND, but also can stop any ongoing clinical study at any point during development. And again, this is based on a safety concern. FDA will always attempt to discuss and resolve the matter with the sponsor before issuing the clinical hold letter. And how does this work? So if the decision of the review team is that the IND is going to be placed on hold, and I have to say that really a hold is a clinical decision. The clinical division places the IND on hold. However, However, every discipline provides a recommendation of safe to proceed or placed on hold. I mentioned that before the 30 days, all the team gets together and the potential hold issues are discussed, and there is an agreement about whether the IND should be placed on hold or not. The decision is clinical, but the different disciplines inform and share their concerns and discuss. And then if the decision is to place an IND on hold, then the division director for the clinical division or someone on his or her behalf will communicate this decision to the sponsor, generally by phone, as a means of really a quick communication, because again, this decision might be done on day 29. So basically by day 30, there is a TCOM with the sponsor, and the communication of a hold is done. The division director will communicate, of the clinical division will communicate the hold to the sponsor, and then will pass if the hold is not clinical, then the issue is not clinical, then the review discipline that identified the safety concern will explain what are the bases for the action, and also what is the information that is needed to address the deficiency. The division director of the clinical division will provide the sponsor a written explanation of the basis for the hold, no later than 30 days after imposition of the clinical hold. So when an IND is placed on hold, the sponsor can respond, should respond to the clinical hold to be able to start a clinical study. The response to the clinical hold must be complete, must be a complete response. This means that if the hold, an IND is placed on hold for 2 or 3 or more reasons or hold issues, the response has to address all the issues identified at the time of the hold. If a complete response is then submitted and the review disciplines go through it and determine that the response is complete, then another 30-day review cycle begins, and the RPM sends the sponsor a new acknowledgement letter. And so again, during these 30 days, the response to clinical hold is revised, and the decision is made on whether to remove the clinical hold, and then the IND is allowed to proceed, or to continue the clinical hold because the safety issues were not resolved. What are the CMC bases for a clinical hold? So an IND is placed on hold from a CMC perspective, basically based on 2 things. If we identify while reviewing the CMC section that human subjects are or would be exposed to an unreasonable and significant risk of illness or injury, or that the IND does not contain sufficient information required in the CFRs 31223 to assess the risk to subjects of the proposed studies. So, or we identify that there is a risk, a reasonable risk, or we don't have sufficient information to assess the risk. And as I mentioned before, FDA may place an IND on hold at any time during development. So, as I mentioned before, so after the IND is started, if it is safe to proceed, the development continues. And during this development, the sponsor can request different meetings with the FDA. There are basically 3 types of meeting: type A, type B, and type C, depending on what is the interaction that the sponsor needs. The type A meetings are meetings to discuss stalled development programs. For example, if an IND holder was placed on hold and needs to understand how to be removed from clinical hold, they can request a type A meeting, because if that hold is not resolved, then the program is stalled. This type A requires a rapid response from the agency, and therefore is scheduled within 30 days of the request, and the mixed-in package, that is all the information that the sponsor submits to the FDA, regarding the information that they want to address in the meeting is submitted with the request. The type B meetings are meetings for milestones. A pre-IND meeting is a type B meeting. The timelines is basically a 60-day. The meeting is scheduled within 60 days of the request, and the meeting package is submitted to the agency no later than 30 days before the sponsor meeting. And the type C is a meeting for all other type of advice that the sponsor might need from FDA, and they are scheduled 75 days after the request. And the meeting package is submitted 30 days before the meeting date. So, as you may see, the type A meeting is a faster response, because the meeting is scheduled within 30 days, and the other meetings have a different timeline. There are different formats for the meetings. The meeting can be held with the sponsor face-to-face. Everybody gets together in a room, or by a teleconference, or also can be written responses. The format can be written responses only. That means that the FDA provides responses to the questions posed by the sponsor, but that there is no additional interaction. When the meetings are held face-to-face, or there is a teleconference, then meeting minutes are going to be submitted from FDA to the sponsor 30 days, maximum 30 days, 30 calendar days after the meeting. So there are FDA developed 4 expedited programs, established 4 expedited programs with and the idea is to facilitate and expedite the availability of drugs and biologics to address an unmet medical need in the treatment of a series of life-threatening condition. These programs are intended to help ensure that therapies for these conditions are approved and available to patients as soon as it can be concluded that the therapy's benefits justify their risk. These programs allow for early attention to drugs that have promise in treating such conditions. Again, all these expedited programs are for drugs, biologics, for serious and life-threatening conditions, and for unmet medical needs. There are basically 4 programs: Fast Track, Designation, Breakthrough Designation, Priority Review, and Accelerated Approval. And you have here the legal basis for each development or designation. So Fast Track Designation and Breakthrough Designation can be requested during the IND, generally should be requested during the IND, and that is why I'm going to discuss them a little bit in more detail. Priority Review, is a request that is submitted with the BLA or NDA application, and basically is an ask for a shorter review clock so that the FDA makes a decision of approval or CR in a shorter time clock. And the accelerated approval is a pathway, an approval pathway, where the approval of a product is based on a surrogate marker of clinical efficacy or a clinical endpoint. So on a fast track designation, a drug can be granted a fast track designation when there is no clinical or clinical data that demonstrate the potential of this drug or biologic to address an unmet medical need. What you gain with a fast track designation? Well, as I mentioned, it can be submitted during the IND, with the original IND or during the IND stage, and the sponsor gains frequent interactions with the review team that will help expedite development and review. They have some benefits at the time of the BLA or NDA submission, like being eligible for rolling review, eligibility for accelerated approval and priority review. And it's important to know that this designation can be rescinded if the qualifying criteria are no longer met. And on the right, you have some data showing some infographics about the number of fast-track designation requests that CDER has received since 1998 to the year 2019. As you can see, these requests have increased with time, and some of the requests are denied because the data are not sufficient to grant this designation, and some are granted. And then here in the bottom, you can see the total fast track designation request over these years, to 1998 to 2019, and the number that were granted versus the number that were denied. And again, this is in CEDAR. With regard to the breakthrough therapy designation, this designation is granted to drugs that show preliminary clinical evidence that indicates substantial improvement for one or more clinical significant endpoints over available therapies. They can be also submitted during the IND, at the time of our original IND or after. And the sponsor has the possibility of have increased frequency of meetings throughout the development so as to facilitate an efficient development program. There is an assigned cross-disciplinary team lead that facilitates an efficient review, coordinates internal and external communications, and also senior management and experienced review staff are involved in breakthrough designation submissions. They also gain some advantages for the time of the submission of the BLA or NDA, like rolling review, and other actions to expedite the review. And again, at the fast track designation, the designation may be rescinded if the qualifying criteria are no longer met. And these are again some data from CIREN and CBER regarding the number of breakthrough designation requests, those that are granted and those that are denied or withdrawn. In the case of CIDER, for example, from 2012 until 2020, there were like 1,553 requests, from which 417 were denied and 368 were granted. In CBER, the number of requests was 162 during the same time frame, with 1,049 and 50 withdrawn. So, as I mentioned, the designation for breakthrough is based on clinical data or non-clinical data for fast track. So basically, the designation is clinical, and that means that the time frame of development is going to be shorter, faster, and so probably the sponsor of a product that receives this kind of expedite development designation will need to pursue a more rapid manufacturing development program to accommodate for the fast pace of the clinical program. And so that's why it's very important that sponsors start conversations with the agency, with the FDA, very early in development, so as to make sure that they can align the clinical development with the CMC development. Basically, because also the requirements for CMC are the same than for drugs that do not have an expedite program designation. However, there are a lot of flexibilities that are applied, and always it's an assessment of risk-benefit. And then these products should also, of course, meet the statutory requirement for approval regarding safety and efficacy. What are the challenges? I started a little bit commenting those in the previous slide, but basically the challenge is based on the need for alignment of the CMC development timelines with those of the clinical development. So the process development and the product characterization have to keep pace with the clinical development. Generally, there is less product and process experience, again, because the time is shorter. There is limited data, limited batches that are manufactured, and limited stability data at the time of submission. So during the IND development stage, when a product has been granted, or program has been granted, or fast track, or mostly breakthrough designation, there has to be a real planning of CMC data collection and development of the manufacturing process to make sure that the program from a CMC perspective is mature enough at the time of licensure or approval. The risk-benefit assessment regarding so the FDA has to do a risk-benefit assessment regarding the risk of probably less CMC information versus the patient benefit. There are like constraints in their review time, and then there are also supply and availability considerations. So during development, the sponsor has to get ready to be able to deliver to the market, to supply the market the drug, to manufacture the drug, and be able to have enough drug to feed the market. What are the lessons learned to really try to align the CMC to the clinical development for this kind of expedited programs? Basically, the sponsor has to, it's critical that the sponsor has a robust product characterization, understands the CQAs of the products and the mechanism of action. It is important to plan the development ahead and to keep it as simple as possible. So, and try to minimize changes. So, for example, because whenever a sponsor does a manufacturing change or a scale up, there is need to do a comparability exercise, and this is very time consuming, and a lot of data has to be generated. It's important to limit the number of drug product formats and formulation changes. Again, to streamline the development, limit the number of initial manufacturing sites that are submitted in a BLA. Maybe it's better to submit a BLA with one site and then add additional manufacturing site post-approval. It's important that the sponsor includes as many lots as possible in the clinical study so that they collect clinical data and they can understand also the variability of their lots and streamline the process qualification. All other things, additional site, additional DP formats, all that can be done post-licensure. Well, I have arrived to the first pause, and please feel free to ask any questions of what we have discussed up to now. Just let me know and we can discuss that.
Cecilia, we haven't received any questions through the chat. I don't know. Everybody can speak in the microphone and ask your questions if you have so far.
Oi, bom dia, vocês estão me ouvindo?
Sim.
Oi, é a Miriam. Eu tenho uma dúvida, não sei se é mais geral, assim. Conforme os documentos vão chegando, né, não só de qualidade, as atualizações dos documentos em geral, como que o FDA se organiza para ir revisando e liberando o parecer, assim, se eles consideram ok ou não?
I think I need translation. I thought I was going to be able to deal with it, but I don't have translation to make sure that I understand the question.
No, okay. And she asked about the new information that FDA's received from sponsor during the development. So how FDA assess the information and how they release their opinion about that update.
Yes.
But I think you are going to cover this. But
I'm going to cover that towards the end of the presentation. But I can basically tell you that as you say, during the IND stage, there are amendments that are sent to the FDA, amendments that for the different disciplines. But for example, for a CNC amendment, we review the amendment. Well, the reviewers review the amendment, and then if there are any kind of doubts or information that is unclear, they can have a communication through information request with the sponsor, and that's it. There is no approval, there is nothing, unless again that information raises a safety concern, and then there is going to be interactions to try to solve it, or there could be a decision during any time of the development that an IND should be placed on hold, and then the clinical investigation should be stopped.
Oi. Está escutando? É a Carla?
Oi, Carla, sim.
Só completando a pergunta, Maria Cecilia, when they have a rolling submission, I mean, the documentation are coming, not everything together, but in a different time. The FDA, do they write a report about it? How do they document that the documentation already sent was okay, and the IND can procedure. How is that documentation for rolling submission?
Yeah, so thank you for bringing that up, because the rolling submission is for initial marketing authorizations. It's not for the IND. The rolling, it means sending complete modules of the submission, but not all the submission together. But this is at the time of a BLA or probably an NDA submission too. Is not during the IND. The IND is a single submission with all the information that the sponsor puts in that IND. And then if there is additional information that the reviewer needs to make their assessment of safety, then an IR, an information request, or a communication with the sponsor will try to address that need of information. Rolling review is not something that occurs in the IND, it's something that occurs for a BLA or NDA. And yes, and so basically it's not an IND thing. The rolling review, I brought it up just because for the expedited programs that ultimately the designation is granted during the IND stage development, they have these benefits for when they are going to submit their NDA or BLA. But there is not such thing of rolling review in an IND.
Yes, but with even though with that rolling submission for the IND, is it documented in somehow, or is it?
Okay, I understand. I think I understand what you say. So yes, and again, from what I used to do, our office had a template for INDs, for new INDs, where and the template basically for commercial INDs followed the same kind of template for a BLA with the different sections where you would put some information and your assessment of the information. And basically, again, this template and the information that we put in the template is related to safety concerns. Certain areas of an IND from a CMC perspective that could be safety concerns, and that we assess the information and make a decision. And so that is documented in a review memo that the primary reviewer performs, and that a team leader oversees and agrees. If there are potential for clinical holds, then that is also documented in the memo with the rationale for why you are recommending from a CMC perspective a hold, and what is the safety concern. Is that what you were asking?
Yes, exactly what I asked.
Oh, okay. Sorry, I misunderstood the question. And then we have a system. There is a system in FDA where these memos are uploaded. So you can access the memos from everybody, from the different products, through this system that is called DARTS. I don't remember what they stand for. I can for sure give that to you. But basically, once you are done, you put it in DART, you put your memo in DART, you sign it electronically, and then it goes to your secondary reviewer TL that signs, and then the recommendation is in the system. And I think it's very important that if the clinical hold is going to be based, for example, on a CMC issue, the clinical division generally would expect that memo is uploaded, so that your decision is uploaded in that system before doing the call, because they want to make sure that you are documenting what the hold is.
Cecilia, I have a question. It's
Sure.
Eloisa. So is this a memo that you are saying for the assessment of the decision exclusively for clinical holds, so when there is a safety concern?
No, it is not. But it is not. But I think it's more important when there is a safety concern. But because it has to document really the rationale. But it also, in the safe to proceed, there is a memo where the rationale for not finding, or at least showing that all the information that is needed is in the IND. That also is, there is a memo generated for both situations. Yes.
Okay, thank you.
At least from a CNC perspective, I'm not sure how other disciplines handle this.
Eu queria perguntar para a Maria Cecilia, não sei se dá para vocês traduzirem para ela, se ela teve experiência na análise desses medicamentos para a COVID, com relação aos dados de qualidade que são apresentados, validação dos métodos utilizados para avaliação da qualidade desses produtos, se ela teve essa experiência no FDA e, assim, quais são a parte... Como que eles avaliaram isso, a bastante falta de dados, né, que tem no processo?
Okay, so Cecilia, there is another question.
Yes.
Asking about if you had the experience on reviewing assessments for COVID drugs or biologics during your term in FDA. This is the first part. And then the second, if you can share, if you are not involved on that before, but if you could share how the FDA is dealing with the CMC part of COVID INDs because of the kind of all new submission, new information coming and the incomplete packages. How the FDA is dealing with that situation.
So unfortunately, I left FDA right before the pandemic. So I really cannot comment on that because I don't know. But I think Camila told me that there is going to be discussions later about that, but Camila may need to confirm. But unfortunately, I cannot comment on that because I was not there during the COVID-19 submissions.
Yeah, actually, we are going to
thank you.
try to bring someone to share the experience about COVID-19 with FDA from another company. From another pharmaceutical, okay?
Thank you, Camila.
You're welcome. Mais alguma pergunta, pessoal? So, okay, I think we can.
Should I continue?
Continue, yeah, we can.
Sure.
Yes,
Yeah.
please.
Thank you, Cecilia.
Sure. So, well, now we are going to go into the format and content of the IND. This may look familiar to you or not, but basically this is the CDD triangle. And the CDD triangle is a kind of, through ICH, the International, Conference of Harmonization, there was an agreement on how to organize the information in a dossier, and basically it was agreed to organize it in 5 modules. And again, this is for dossiers for marketing applications. However, INDs for commercial INDs are expected to have this kind of organization. And basically, there are, as I said, 5 modules. The module 1 will have administrative and prescribing information, including, for example, labeling. Let me move this. So the module 2 will have overviews and summaries, and this could be quite valuable for reviewers. For example, if I am a CMC reviewer and I need to understand something about the toxicology studies, I will not go into the reports, but I will be able to look at the summaries and have a sense and probably get an answer to my question. And the same with any other kind of the information that is outside the discipline that a reviewer reviews. So also they are very, very useful for the cases in which an IND is not reviewed by the same reviewer during the development. The idea in FDA, or at least in the office I belonged to, was that the same reviewer would follow the IND since the original submission, probably until the BLA and post-marketing. However, sometimes because of workload or because the reviewers leave the agency or for different reasons, that is not possible. And in that case, this quality overview, overall summaries could really give a sense of what is the stage of development, what is the IND, the product, and some general information for a new CMC reviewer that has to review now an amendment or maybe even the BLA. Then there is module 3, in which the information about quality is included, and this information should cover drug substance, drug product, placebo, facilities, advantageous agent evaluation. And module 4 will contain all the non-clinical study reports, and module 5 will contain all the clinical study reports. So and as I have mentioned, we are going to focus now on the content on the IND in the module 3. What would be the module 3? That is quality. And I think I should also mention that, again, this organization is for a marketing application. So for an original IND, the amount of information that a sponsor will submit is going to be much less than the information that is going to be included in a marketing application. And that is because really the IND is, there is some knowledge that the sponsor has about the product that ultimately is sufficient to know that they are not going to pose unreasonable risk to patients or subjects, but ultimately during development, the sponsor is going to collect information about the product, about the process, understand and refine the process to scale up. So really the amount of information increases with development. The content of an original IND is much less than the content of an original BLA, and that it's because obviously a lot of data are collected during IND development. So what are the requirements for the CMC requirements for an IND? An IND has to must have information for the drug substance, the drug product, the placebo, labels and labeling, environmental assessment, and also a brief description of the drug substance and the formulation, including the structure and formula, and this information should be included in the investigator's brochure. As outlined in the CFRs, the CMC content in an IND has a graded nature. So for each phase of the investigation, there should be sufficient information to assure identity, quality, purity, and the amount of information will vary with the phase of investigation, the proposed duration of the investigation, the dosage form, and the amount of information otherwise available. Again, this is what I was mentioning before. You need sufficient information to support safety and then efficacy, but really this will vary with the phase of development. So how do an FDA reviewer look at an application and review this application? And basically, the primary objective in reviewing an IND in all phases of the investigation is to assure the safety and rights of subjects. I cannot emphasize this enough. An original IND is about safety, and then during all the IND development is safety and effectiveness for phase 2 and phase 3. And this is further kind of elaborated in this guidance for industry, for the context and format for an IND for phase 1, that says that the emphasis in an initial phase 1 CMC submission should generally be placed on providing information that will allow evaluation of the safety of subjects, the identification of a safety concern, or insufficient data to make an evaluation of the safety is the only basis for a clinical hold based on CMC. And again, the data required to assess the safety of the drug product at each phase of development is product specific. So let's go now a little bit in more depth in the CMC information inside the CMC package. So I mentioned that there has to be drug substance information. What is drug substance? Drug substance is an active ingredient that is intended for the incorporation into a finished dosage form. That means the statutory definition of a drug that is intended to furnish pharmacological activity or direct effect in the diagnosis, cure, mitigation, treatment, or prevention of a disease, or to affect the structure or any function of the body. So drug substance is the active ingredient, and it's important that it's not to be used in the human subjects. So you don't deliver to subject the drug substance, you deliver the drug product. What is the information required? Description of the physical, chemical, or biological characteristics of the drug substance, the name and address of the manufacturer, a description of the method, the manufacturing process, the method of preparation and synthesis, the limits and analytical methods used to ensure the identity, strength, quality, and purity, a description of the methods used, and information to support stability during the toxicology and clinical study, and also the batch analysis for the clinical lot and the toxicology material. This slide basically shows a typical manufacturing, drug substance manufacturing process for, at least for a biologic, for a biotech product. This process can be divided into an upstream process and a downstream process. For the upstream process starts with a vial of the cell bank that is subjected to expansion steps and until enough material is obtained to purify the product of interest. And then even the cells are harvested or the supernatant, and they are used for the downstream process that basically is aimed to isolate and purify the product to a target level of purity and quality. And basically this is done through a series of purification and concentration steps that allow, again, to obtain a product with a certain quality and purity. So for biotech products, there are different cell substrates that can be used: bacterial, mammalian cells, yeast, plant, maybe the pancreas of a pig, eggs. At the time of the IND submission, the cell bank has already been developed, and it has been tested for identity, purity, and stability. And it's very important that the cell substrate, when they are of biological origin, like all these substrates, they are tested for adventitious agents. This could be viruses, TSC, bacteria, mycoplasma, fungi. And this is very important because these cell banks, the cell bank can be the potential source of adventitious agents contamination, and also because they are an optimal environment for the growth of adventitious agents that can be introduced into the manufacturing process through the raw materials or through the environment. So because, again, there is the potential for a viral safety concerns, and because the potential presence of an adventitious agent in a product is a safety concern, then the original IND for a phase 1 has to have information to support the safety of the product from a viral perspective, viral safety perspective. And this includes information about for the testing of the cell banks for the absence of adventitious agent that could be endogenous or not endogenous, through the control of raw materials. So the IND should have a list of raw materials of animal origin and what is their source, if it is an animal of biological origin, and they could be from animal origin, plant, or they are recombinant. There should be information of the country where the animal-derived materials are sourced, or letters from suppliers verifying that the animals-derived materials came from BSE free countries. And then there is also need for testing of the unprocessed bulk for absence of adventitious agents. And this is because the unprocessed bulk is the latest state of the fermentation and is the place where there is the highest probability of finding an adventitious agent if it is there. So again, testing for cell bank, control of the raw materials, testing for the unprocessed bulk. And then there should be tests for retroviral-like particles in the case of CHO or NSO cultures. So one part is the testing. The other part to support viral safety is the result for that the sponsor has to include in the IND result for viral clearance studies. And these studies are aimed to demonstrate the capacity of the manufacturing process to clear and inactivate viruses. So if there happens to be the introduction of the virus in the manufacturing process, the manufacturing process would be able to remove or inactivate these viruses. And those are the data that should be submitted in the IND. There should be at least 2 orthogonal, robust verification steps in the downstream process. When I refer to orthogonal, I mean different method of inactivation or removal. For example, pH inactivation is a very robust inactivation virus, and filtration is a very robust removal step. These viral clearance studies are not performed at full scale. You are not going to introduce viruses in your facility. They are performed at small scale that mimic the step that you want to test for viral clearance. And so it's important that in the IND submission, the sponsor demonstrates or provides some summary data that demonstrate that the small scale model that they are using to test viral clearance is representative of the full scale manufacturing step. And then the idea is that the sponsor calculates a safety factor. And this means basically having into consideration the capacity of the steps, the viral clearance steps to remove or inactivate viruses, and calculate what is the potential possibility of adventitious or viral particles per doses. And the expectation is that at this point, as per ICH Q5A, the expectation is that this calculation leads to less than 1 dose every 1 particle every 1 million doses. So I'm going to share with you a case study. This case study that ultimately shows some deficiencies found with regard to viral safety in an original IND. This was an original IND for a recombinant protein that was produced in CHO cells. The sponsor evaluated 2 steps for viral clearance and then calculated the log reduction factor. This means that how much each step is able to reduce the presence of viruses. And so when the sponsor provided this data, really this value was too low to ensure that the calculation of the safety factor, the calculation of how many particles per doses so that basically they couldn't get to the 1 particle to 1 million doses, and therefore there was not sufficient assurance of safety. And then also the sponsor didn't provide information to support that the small scale model that they were using to do the viral clearance studies was representative of the full scale study. The IND was placed on hold because there was insufficient information to demonstrate a robust viral clearance capability of the downstream process and therefore an acceptable safety margin. And then to be removed from the hold, the sponsor repeated the viral clearance studies, evaluating additional steps so that this way they could show that the manufacturing process had sufficient capability to remove or inactivate viruses that could enter the process. then it was able to really reach a viral safety margin that was acceptable, and also provided information to support that the scaled-down model was representative of the full-scale one, full-scale model. So what are the data that are expected for the upstream to support or to describe the upstream manufacturing process? A list of the materials and their sources, and we have discussed this already. A description of the fermentation and the harvesting processes, including controls and limits, the address and the description where the manufacturing site is, the testing of the unprocessed bulk, and we mentioned this, and also the hold times. Many times during the manufacturing process, there are holds so that the process is stopped, and so this had to be somehow controlled for bioburden and endotoxin, for example. So as development proceeds, the sponsor is expected to establish in-process controls and set alerts and action limits to better control their manufacturing process and be able to consistently deliver a product of the expected quality. Regarding the downstream manufacturing process, information describing the purification process, including control limits, including controls and limits, has to be included in the IND. Information about the removal and the characterization of process-related impurities like host cell DNA, host cell proteins, antibiotic induction agents, detergents that could be used in the fermentation, and also testing for adventitious agents. And we had also discussed this part. And also information about regarding the removal and all the characterization of the product-related species. Are there aggregates, clear products, charge variants, aggregates again, and develop also a method to test and compare impurities across the lots. With regards to drug substance characterization, information to characterize the physicochemical properties of the drug substance, including in identity, primary structure, high-order structure, post-translational modifications, biological activity, should be included in the IND. Also, quantity, purities, impurities, and contaminants, and the data should be tabular and also primary data. So not only the numbers, but also maybe some images from gels or chromatograph profiles are very helpful. So in order to enter a clinical study or clinical trial, the product does not need to be pure or fully characterized. It's not, because I mean, early in development, you don't have that information, or you don't understand your product and your process enough. However, what is really key and critical is that the possibility or the capability to compare product quality between lots, so that the sponsor can apply the safety information that they collected during the preclinical studies and the early clinical studies, and the dose finding data in the early clinical studies, they can really relate it to the later clinical phases, 2 and 3. And this includes potency. And so it's really important to really be able to establish this comparison between lots, early and late lots, that the sponsor develops and identified as early as possible relevant release, characterization, and stability assays that are sensitive and they are precise. And the assays don't need to be validated prior to licensure. So during the IND stage, you don't need assays to be validated. You need assays that are suitable for their intended purpose, so that they are going to be able to provide useful and relevant information and reliable information. So elaborating a little bit on the previous slide, it is to establish the safety. So the sponsor will do some toxicology studies before submitting the IND. They will manufacture a toxicology lot, and they will test the toxicology and pharmacological activity of the drug in animals. They are going to then manufacture a clinical lot that is going to be the lot used in the proposed clinical study. In most of the cases, the toxicology lot or the lot used in the toxicology studies and that proposed for the clinical study are different. And there are some cases that maybe there is one lot that is used for both ends, for the tox studies and for the proposed clinical study. In that case, of course, the lot has to be manufactured under GMP. Many times when there is a tox lot and a proposed clinical lot, the tox lot might not be manufactured in the GMP conditions. The clinical lot has to. So to really breach the safety information collected in animals using the toxicology lot and assure the safety of the proposed clinical lot, the sponsor needs to establish a comparability. And this is explained in this FDA guidance for industry about the content and format for INDs for phase 1, that basically says that the sponsor should be able to relate the drug product being proposed for use in a clinical study to the drug product used in the animal toxicology studies that support the safety of the proposed clinical study. This is a very important part of our initial IND submission. The comparability assessment or data to support that the toxicology lot and the proposed clinical lot are comparable. So this is the question. Is the non-clinical material representative of the clinical material? So, and in doing so, this activity, the question is whether there have been manufacturing changes in the manufacture of the tox lot and the clinical lot, and whether those changes are likely to impact safety. So are the manufacturing process for the tox and the clinical lot comparable? Based on this assessment, then it's important to do a risk-based comparability assessment that ultimately understands what are the key aspects of the product that need to be evaluated to support this comparability of the tox and the proposed clinical lot. In the data, expected is tabular and also primary comparability data, and I will show you later a clinical an example, a case study that shows the importance of having primary data to assess the comparability of the tox and the clinical lot. And again, this comparability, analytical comparability should focus on quality attributes that as they relate to safety. So process and product related impurities, potency. This slide is not for you to read every detail. It's basically to show you that there is a huge variety of methods that could use to assess key aspects of a drug substance, like safety, purity, impurities, identity, potency, protein contents. There are some methods that are more suited for small molecules like infrared absorption, and some others that are more useful for biologics. In the case of biologics, potency has to be part of the release testing, and different formats of these assays could be used. There could be animal assays that are barely used these days, cell-based assays, reporter assays, ligand binding. For the case of small molecules, potency is measured more for the protein content. The release testing. So you have several aspects of the characterization of your drug substance, and you use different and the sponsor uses different methods to really characterize the drug substance. So, but a subset of these characterization tests are really part of the release testing. The release testing has a subset of tests that measure key features of the drug substance, and also that these methods have to be able to be reliably performed in the QC lab under routine basis. So this is a subset of assays that are chosen to confirm the quality of a product. So when there is understanding of what are the quality attributes of a product that really need to be monitored and controlled to ensure the quality of the product, that is what goes into the release assays and the release testing. And so we talk about release specifications, and when we talk about release specifications have three components, include the quality attribute, like for example, identity or purity, the analytical procedure to assess this attribute, like, I don't know, reverse phase HPLC or cation exchange or different assays for purity, and then the acceptance criteria. There are numerical limits, ranges, or other criteria. These specifications can be broader early in development, and then as manufacturing, as experience and development experience increases, then the sponsor can tighten them. It's important that original INDs do not have acceptance criteria report results, because this will not ensure that the next lot that the sponsor manufactures will have a quality that is comparable to the lot that we see in the original IND. So release testing should include testing of identity using an assay that is able to uniquely identify the protein of interest. They should control for purity and impurities, for potency, for quantity, and also for microbial safety. Now, what is a drug product? So we talk about the drug substance. Now we are going to talk about the drug product. What is a drug product? It's a finished dosage form that contains a drug substance and generally in association with one or more other ingredients. The drug product is intended for use in human subjects, and there is need for sterility assurance when the drug products are for parenteral use. What is the information required for drug product? The components used in the manufacture, the analytical methods and limits, description of the manufacturing process and the packaging, information to support the stability during toxicology and clinical studies, the name and address of the manufacturers, a brief description of the composition, manufacture, and control of a placebo if the proposed clinical study is a placebo-controlled one, and copies of labels and labeling to be provided to each investigator. So this is a kind of, it's a cartoon that shows probably a typical drug product manufacturing process. In the case that the manufacturing process for drug product starts with the drug substance, in the case that the drug product is is frozen, that is generally the case, then the drug substance is thawed. Sometimes the sponsor may pull different drug substance lot to manufacture a single drug product lot. If that is the case, then thawing and pulling will be the first step. In the case that the drug substance and the drug product have different formulation, then the drug substance has to be formulated into the drug product formulation, generally by diafiltration and ultracentrifugation and buffer exchange. And then this formulated drug substance is filtered and then filled into the final container. That generally for original INDs are vials because are the easier container closure. If lyophilized, then lyophilization takes place. All the vials are going to be visual inspected, labeling, packaging, and then shipment and storage. Drug substance and drug product are generally manufactured in different sites. Some of these steps, as I mentioned, may not be needed. For example, formulation might not be needed if the drug substance has the same formulation as the drug product. So in that case, the manufacturing process for the drug product would be basically thawing and filling of the drug substance. Many times, pooling is not also part of the manufacturing process, and one drug substance lot gives rise to one drug product lot. And lyophilization generally is not seen in original INDs because really is a complicated process. In most of the cases, original INDs are liquids in vials. Drug product release testing, some of the testing that we have discussed for drug substance, and also some drug product specific tests like sterility, particulate matter, volume and weight in the container, residual moisture or reconstitution time for lyophilized products, excipient concentrations, and also an assessment of those impurities that may form or be introduced during the drug product manufacturing and storage. The it is a requirement as per the CFRs for the IND to include stability data. Stability data are needed in all phases of the IND, and basically there should be sufficient information to support the stability of the drug substance and the drug product during the toxicology studies and the plant clinical studies. So an IND submission should have sufficient stability data to support the stability of the product during the for the duration of the clinical studies. Some considerations about stability testing. It's important for sponsors to identify stability indicating tests that confirm the stability of the drug substance and drug product under the recommended storage conditions. The submission should provide data or should include data to support the stability of the product for the duration of the toxicology and clinical studies. We have already mentioned this. There should be stability protocols under recommended accelerated and stress conditions provided in the IND submission. This is basically what is the test schedules and what are the test methods that the sponsor is going to use over time to generate data that support the stability of the material they are using in the clinical and in the they have used in the toxicology studies. For these stability studies, the drug substance and drug product have to be stored in containers that are representative of the containers used at full scale. So, so that the stability data that are collected are really representative of the stability of the full-scale material. It's very important that the sponsors start collecting stability data as early as possible, so therefore it's important that they place the toxicology and the first GMP lot on stability. And stability data should be collected through development. It's important also that the sponsors does assesses the evaluation, assesses the stability of the process under accelerated and stress conditions, because these will give information about the degradation pathways of the products and also help identify which are those assays that are able to detect changes in the stability profile of the product. And one of the aspects that is very important for drug product stability is in-use stability. The IND should include data that support that the product is stable during handling and administration, and that the product is compatible with the delivery system. So let's think you have a product that is filled in a vial, but ultimately it's going to be administered to patients through an IV infusion. So that means that the vial has to be opened, sterilely breached, then diluted in some diluent, put in the bag, and may stay in the bag for a little while before and while the infusion occurs. So it's important to provide data that supports that the product is stable during all that process and using the conditions and the materials that are going to use in the clinical setting. And this is especially important for high potent products that are administered at very low doses. There should be data that support adequate recovery of the product from the infusion bag. And I have a case study here that shows an IND that was placed on hold because the recovery data was deficient. But what I think I want to explain clearly here is that when you have the rationale for and the importance of this as it relates to safety, when you have a very potent product, and probably the starting doses for clinical studies are going to be very low, and so it's important that the sponsor confirms that the intended dose is received by the patient, and the patient or the subject, the clinical subject, is not receiving less. And this is because when you do the safety readout of that dose to inform your dose escalation scheme, then you need to be sure that the safety information that you are collecting is for the dose that you intended to deliver and not for a lower dose, because then if you think that a certain dose is safe, but you really were assessing a lower one, then when you do the escalation, you may have safety adverse events. So this IND was an original IND for an antibody drug conjugate that was administered by IV infusion. The sponsor proposed doses from 0.01 to 1 milligram per kilo, and provided also recovery data, simulated recovery data for the low and high doses in the 2 diluents that they were planning to use, saline and 5% dextrose. And they used the administration set that they were planning to use in the clinical setting. So this data here show the percentage recovery. And as you may see, if you focus on the saline inline filter data, the sponsor tested time 0, time 4, and time 24 hours. Basically, the amount of drug before the inline filter and after the inline filter for each time point. And what the reviewers saw is that in saline, at the lowest dose, the recovery is very low. Around 43% of the drug is recovered after the filter, and so this is not acceptable. And so the sponsor, so the issue was that there was no assurance that the patients will be receiving the intended dose, especially at lower doses in saline, and so the sponsor would collect really safety information that would not be reliable, and then it inappropriately will inform the dose escalation. So to address the deficiency, the sponsor was asked to provide data to support the product recovery using the proposed administration materials and diluent, identify the factors contributing to the low protein recovery, and implement a strategy to ensure consistent protein recovery at doses used in the clinical study. Another case study, this relates to stability data. This was an original IND submission for a recombinant protein. The sponsor provided stability data for the toxicology lot and not for the proposed clinical lot. And the problem was that the toxicology lot showed a time-dependent increase in impurities by reverse phase HPLC, so the 6 months period of storage and the recommended storage conditions of -20 centigrades. So the IND was placed on hold because there was no assurance that the proposed clinical load would remain stable throughout the planned duration of the clinical study. To be removed from the clinical hold, the sponsor provides sufficient real-time stability data, including primary data, for the clinical load to ensure stability during the clinical study. And I think this is the last case study. This is an original IND submission for a recombinant protein. And this was a hold IND due to inadequate data to support comparability of the toxicology and the clinical lot. So the sponsor provided release results for the toxicology and clinical material, and all these results met the release acceptance criteria, but only tabular data were provided in the IND. So the reviewer requested high quality primary data like gels, chromatograms for the toxicology and the clinical material. So when these data were received, there were differences in identified in the purity profile by the SDS page, for purity. There were high and low molecular weight bands in the clinical lot that were not observed in the tox lot, and these were not detected by Western blot, meaning that they were not product related. So the IND was placed on hold because there was insufficient data to support comparability between the toxicology and the clinical material. And as you can see that here, the tabular data was within specs. So, for example, I don't know, purity more than 90%. So it will pass. But when we asked for the primary data, we saw these different impurities in the clinical lot. And so What I want to stress here is the importance of looking at primary data. So to be removed from whole, the sponsor needed to explain the nature of the high and low molecular weight bands observed in the clinical material and their potential impact on patient safety. Another aspect of another safety concern for an original IND and also for an IND during development is immunogenicity. So the development of anti-drug antibodies. Anti-drug antibodies can be a safety concern, and so they need to be assessed and measured through development. In the case of very high-risk products, there might be need to have assays in place for phase 1, and but otherwise the assays should be in place towards phase 2. In the absence of a qualified assay, then the sponsor should collect sera during appropriate times of the clinical study and bank them so that when they have assays to test for immunogenicity, they can test the presence of antibodies in these samples for early clinical studies. During development, the sponsor has to develop and validate immunogenicity assays to assess binding antibodies and also neutralizing antibodies. And the immunogenicity rates for a product are going to be reported in the label. The biggest immunogenicity concern is the potential of cross-reactivity of an antibody response with a non-redundant endogenous protein. And this will deal to a deficiency syndrome. And an example of this is erythropoietin. So this is basically the highest concern. And there are some products that are high risk and some others that are not. But anyway, immunogenicity has to be tested, assessed. Common CMC hold issues that the IND does not have clinical lot manufactured, that there is insufficient characterization of the cell banks or data to support viral clearance, or there is insufficient data to support comparability of the toxicology and proposed clinical lots, and we saw an example of that. Lack of potency assay, inadequate specifications for release and stability, for example, report results. There are not numerical limits, and this is not sufficient to really ensure that the next lot of material that the sponsor is going to manufacture will be comparable to the previous materials and to those used in the toxicology studies. Lack of information of raw materials of animal origin or insufficient stability data. So I have 2 polls here that I'm going to kind of walk through and then tell you what is the right answer. So this is about what is not a whole issue for a phase 1. So the first one is the lack of primary data to support comparability of toxicology and clinical law. And we have discussed that these data are needed, that this is part of the safety evaluation. So that is not a correct answer. Release and stability assays are not validated. We have discussed that really stability data assays for release and stability testing do not need to be validated during IND. The validation has to be, there is need for validated assays pre-licensure. Testing of the cell banks is a requirement for an IND. And lack of potency assay is also a hold issue. So basically the response here is number B. The second poll is what is the reason to put an IND on clinical hold? And this is quite clear, hopefully, and because we have mentioned several times that a safety is what really that a clinical hold is based on safety concerns. It's not about efficacy, at least not during the early phases, and safety and a safety concern is and insufficient information to assess risk to subject is a safety concern. So basically here A and C are the correct answers. Okay, second and last thought for any questions that I could answer.
Eu tenho perguntas. Eu não me apresentei da outra vez. Meu nome é Mariela. Eu tenho umas 2 perguntas, eu não sei, 3 perguntas, se eu faço direto ou faço uma de cada vez. O que vocês acham melhor?
Acho melhor uma de cada vez, Mariela, por favor.
Tá, a primeira é sobre o clearance viral. que ela falou que tem que ter 2 métodos, o adequado, né? Seria ter 2 métodos ortogonais para estar tendo a inativação viral na vacina, por exemplo. Se a empresa só demonstrar que ela inativou o vírus, não é necessário ter esses 2 métodos, ou o que ela pensa sobre isso? Se é sempre recomendável ter 2 métodos?
Okay. So, Cecilia, there is a question regarding viral clearance. So you explained it that the ideal is to have 2 orthogonal methods. But the question is, if the company, if the sponsor has provided in the dossier for the clinical trial application that the virus is inactivated, so is this 2 methods mandatory in terms of FDA perspective? Or there could be the proof of viral inactivation could be accepted?
So the expectation for an original IND is 2 orthogonal methods. And basically, the idea is also that you expect a certain clearance level, and it's highly improbable that one step would give it. But the expectations are 2 orthogonal methods. And basically 2 orthogonal methods for 2 products, 2 orthogonal methods, and a representative virus, so a retrovirus that ultimately would represent the retroviral particles in the CHO cells. So for the licensure, you may need to test more other viruses, but for original IND of a CHO, 2 methods, 2 orthogonal methods, and 1 virus could be sufficient.
Ok. Tá respondida?
Tá sim, obrigada.
De nada.
A outra é que ela comentou que não é necessária ter validação dos ensaios que são utilizados para avaliar a qualidade do IND. Então, o que eles usam para dizer que aquele ensaio, assim, que avaliação que eles fazem para dizer que o ensaio é apropriado, né, para o uso que ele está se propondo?
Ok.
Por exemplo, ela colocou assim, até que ela compara lotes pré-clínicos e clínicos para ver se ela faz essa comparação. Aí como que, se o ensaio não é validado, como que daí ela garante que essa comparação foi fiel, sabe? Eu queria saber o que eles usam para avaliar, então, o ensaio.
Okay. Cecilia, there is a question regarding the analytical validation, for assays. So ANVISA is questioning regarding how does FDA make sure that it's reliable method? Because if it's not validated yet in the IND, so how FDA chooses the best assays that should be performed or accept the preliminary assays results, and how this comparability, you mentioned that there is a comparability between non-clinical batches with some clinical batches, but if you could explain us a little bit better, so how FDA makes this decision, it would be very good to clarify the grounds of decision making.
Yes, so I think it's a great question because a formal validation exercise is not expected for an original IND. But there needs to be data that support that the assay is suitable for its intended purpose, what means basically that it has acceptable precision and accuracy probably to really deliver reliable or relevant results. And this is also part of the specifications. So when you set specifications, I mentioned that the specifications can be broad at the beginning. You don't know the variability of the manufacturing process, but you may have a sense of a variability of your method. And so, for example, if you are going to have an ELISA to test for binding, and the acceptance criteria for a release ELISA is 50%, plus minus 50%, that would not be acceptable because it's known that an ELISA variability could be maximum 20%. So ultimately, there are like the variability of some methods is kind of understood, and and there are some, and then there is also control through the specifications. I don't know, that answered the question?
I think you said that you asked. Vou falar em português, tá? Eu acho que ela falou assim que ela vai pedir alguns pontos da validação, só não vai pedir a validação completa. E eu não sei se eu queria só ver se eu entendi corretamente. E ela falou assim que tem alguns ensaios que já é conhecida a variabilidade deles, aí a gente só tem que ver se está de acordo com essa variabilidade. Se você puder só confirmar com ela se é isso.
Sim, okay. Cecilia, so it's just to confirm if we understood correctly your clarification regarding like known methods, like ELISA, as you mentioned, when it's already known in the literature, what is the variance? And so it's just to confirm if this is the case of we can, ANVISA can make the comments in on this because of a known method. Right, so this is the first part of the question, and then second is just to validate the understanding of, okay, so for the IND is not needed any formal methodology validation, right? But it's required that the sponsor can present this, what is acceptable for the precision of the method, and also to in the justification of preliminary specifications. So it's just a double check if we understood very well your comment.
That is the concept, and basically the sponsor needs to provide data on the method they are going to use and the information that they have at the time. But again, some data to support that the assays are acceptable. Again, it's not going to be a formal validation, but it's going to be some qualification of the assays that show that they are suitable.
Ok, então let me just translate. Então, só para traduzir aqui, o que ela está falando é que realmente não tem nenhuma validação de métodos analíticos durante o processo de IND, mas a apresentação de que o método já tem as características dele em termos de precisão, acurácia e outros componentes dessa verificação técnica, e então seria aceitável apenas para o FDA, né, na análise FDA, essa análise não validada ainda, mas que num futuro prévio, no momento da submissão do registro, aí sim teria que fazer a validação completa dos métodos, tá? Ficou melhor? Ou
ficou,
não,
entendi bem.
Perfeito. Muito obrigada, viu, pela pergunta. Foi ótima pergunta.
A última é sobre a consistência dos lotes. Parece que eu li num dos slides dela que é necessário a consistência dos lotes para medicamento investigacional. Eu queria confirmar isso, se é necessário, e como que... Aí, mais uma vez, né, a minha dúvida, né, se os ensaios não estão validados, como que se garante, né, a consistência? Mas isso acho que eu já entendi. Só para comentar, assim, com relação para ela comentar sobre a consistência necessária dos lotes nessa fase do ensaio clínico.
Tá bom. Cecilia, it's regarding some past slides you mentioned about batch consistency. So again, if the methods are not totally validated at this moment of the development, so how FDA will look at those batches provided by the sponsor in terms of what FDA would consider for this review?
So basically, the importance for this lot-to-lot consistency are the specifications. So with an original IND, you kind of agree that the specifications that are in place at that time, with the acceptance criteria, the method, and the attribute that they are testing, are acceptable to and give assurance that the next lot that the sponsor is going to manufacture at least is going to be within those limits that are established based on the experience that you might have, that probably is 2 batches, the tox and the clinical lot. But you see some variability. You understand the variability of the assays. Probably there is information from the sponsor, and the sponsor proposed some limits. And so that is why it's important not to have report results in the specifications as an acceptance criteria, because a report for most of the critical assays, because a report result will not ensure that the next lot is going to have the same characteristics as the ones that you have. So through the specifications and the acceptance limits criteria in the specifications, you give some assurance that the next lot is going to fall within those limits, and therefore is going to be comparable to the talks and the proposed clinical. And that's why when you understand more the variability of your manufacturing process, of your methods, of that attribute in the product, then you might tighten those. And that is why it's very important that those data are collected early, and then that you can bridge them to the later stages, so that also you can understand the safety that you collected early and the doses, and have that information to inform the later stage. But basically, that's why established specifications, despite the fact that they are going to be kind of broad originally in the original IND and early in development, at least they give you some frame for where the different attributes should land.
Ok.
Muito obrigada.
Ficou claro? Tá tudo bem?
Ficou, tudo
Então
certo.
tá bom.
Obrigada.
Muito bom, obrigada. Eu que agradeço. Alguma outra questão para a Cecília? Que a gente já está... pessoal, só em termos de tempo, eu acho que falta muito pouco. Camila, só dá um help aqui. Quantos slides faltam?
Faltam mais ou menos uns menos de 10.
Ah, perfeito. Então, gente, desculpas por a gente estar estendendo um pouquinho, mas acho que está sendo bem valioso e a gente vai continuar aqui o processo, tá bom? O esclarecimento da Cecília nos próximos 8 a 10 slides no máximo. Cecilia, I was just telling the team that we already passed 5 minutes. No problem. So it's good. I think it's been a wonderful moment to make all these knowledge checks with you. So if you can please continue.
Sure.
Heloísa.
Oi, desculpa. Pode
aqui
falar.
É a Adriane, da COPEC. Eu tenho 2 perguntas, são bem rápidas.
Ah, tudo bem, pode fazer.
Eu vou fazer separadamente também.
Tá.
A primeira pergunta é em relação ao banco de células. Para esses casos de produtos que têm esse desenvolvimento acelerado, como, por exemplo, medicamentos para COVID-19, em alguns casos, a caracterização do banco de células não é completa. Em alguns casos, a caracterização ainda é no nível de bancada. Então, assim, eu queria saber da Maria Cecilia o que seria considerado como risco para esses casos em que a caracterização do banco de células ainda é tão inicial e o IND ele é aprovado com esse tipo de dado.
Tá. Vou fazer primeiro a pergunta, tá bom? So,
Certo.
Cecilia, we have a question coming from cell banks. Okay, so in the context of COVID-19 studies, so is FDA okay to receive incomplete cell bank characterization? And because what ANVISA is saying is that they want to hear what would be the risks of receiving just a cell bank characterization as a basic risk at the basic research level. So like in academic level of the research, and what would be the risks involved on having such a cell bank with just a preliminary characterization in terms of a study for the making the next steps for development?
Yes. So the first thing is that I really cannot comment on what would be the approach of FDA. I cannot comment on that. But it's always a risk assessment. It depends on what is the substrate, what do you know about it, what is the testing that you have, and what is the testing that you don't have. But basically, is that for CHO cells, you don't need for an original IND a complete testing. You don't need the animal testing, for example, for the cell bank. You are going to have it then for licensure. So there are some of the testings that might, and again, as I mentioned before, for a BLA submission, you would expect clearance evaluation of the manufacturing process with 4 viruses or 3 viruses. For an original IND, for example, for CHO, with a retrovirus, with a model retrovirus would be sufficient to open an IND. So despite the fact of COVID-19 that I really cannot comment, but depending on which is the cell, how much experience there exists with that cell, what are the assays that ultimately can be postponed to later in development or for licensure.
Ficou bem, Adriane?
Sim, obrigada.
De nada,
Ficou bem claro. A segunda pergunta é como que os dados da análise que é feita no IND, como que esses dados eles são aproveitados no momento da avaliação para registro, né, do NDA ou do BLA? Como que é feita essa ponte?
Uhum. Ok, muito boa pergunta, obrigada. Cecília, we are receiving question, how does this bridge of the IND data that has been put all together during the IND to the marketing applications of an NDA or BLA. So how does FDA make that bridge of information? So how is the workflow? If you can comment around that, please.
So I'm not sure. I mean, I'll tell you what I think, and I'm not sure whether it's going to answer the question. I think I meant, so the bridge, the BLA application is a new application. It's not that it built, I mean, the information it builds up on the IND, but the IND, the BLA will include all the information in a package that will include, of course, data generated during the IND. There is a section about development, but it's a separate submission, and the separate that it's a standalone submission. So that, for example, as I think I mentioned, a CMC reviewer could be the reviewer of the IND or pieces of the IND, or some submissions of the IND, but then might not be the reviewer for the BLA. And really, a reviewer for a BLA that hasn't seen the product during IND is able, I mean, it takes more time because it has less knowledge about the product, because it hasn't had the product during IND stage, but ultimately all the information is in the BLA. All the information is there. So it's a separate package that, of course, has information from the IND.
So basically, so yes, we understood that is a totally separate package, is a new dossier that was put together for the marketing application. But internally in the FDA, so what you mean is that it could be totally a different reviewer without any experience during the development program, but it could also be somebody that was the reviewer during the IND and could be assigned for the marketing application, right?
That is generally the idea. It's just that sometimes it's difficult to do, but that is the idea. And when the workload was lower, generally a product stayed with the same reviewer or same team until post-marketing.
Okay. So, but like in the internal procedures, so there is no establishment of per internal FDA procedures that one area should communicate to the other. So you do the IND review, and then once the company decides to make the marketing application, it is a totally new submission, and it will go to the area of the expertise, or to the office, or CBIR or CBIR, right?
Well, if it is biotechnology products, it's going to be OBP in CBIR. If they are some blood biologics, it would go to CBIR. And if it is a small molecule, it will go to CDIR, and a different office than OBP or NDP.
Okay.
But again, and I think just to make sure that I said this and I was clear, they are going to be review teams for each discipline in a BLA or in an IND. And so, and all these reviews are coordinated, and there are discussions, and there is always, because things, CMC impact clinical, and there are like all these discussions occur constantly during the review clock for an original IND, during the IND development, and during BLA or NDA submission.
Ok, great. Ficou claro, Adriane?
Sim, obrigada.
Conexão, mas não acaba tendo. Tá ótimo, obrigada. Cecilia, I think we can proceed, please.
Perfect. So this is the last piece, and it's to touch bases a little bit on the maintenance of the IND. So as development proceeds, and I think I mentioned this already, there is an increase in the level of control of the manufacturing process. There is a more clear identification of the critical process parameters, and there is establishment of process controls with alert and action limits and the critical hold times. And the expectation, or the most, the optimal situation is to establish the manufacturing process prior to the manufacture of the phase 3 material. And then during this development, the sponsor should submit amendments to the IND every time significant changes in the manufacture are incorporated, and these amendments should be sent to the agency in a timely manner. So what is an information amendment? In an information amendment, the sponsor should include any information that is not in the scope of a protocol or of a clinical protocol or an IND safety report or an annual report, and it's about new technical information, for example, CMC or tox. CMC amendments can be submitted at any time during development, and the amount and type of information that to include an amendment will depend on where you are in the phase of investigation, the duration of the investigation, and always has to be submitted if it may impact safety. FDA recognizes that modifications to the methods of preparation of new drugs and dosages and changes in the dosages themselves are likely to occur during the investigation, during the progress of the investigation. So, for example, there is need to changes in the scale or in the production of the product from small pilot scale clinical studies to bigger late studies and for the pivotal studies. And so the sponsor should submit information amendments to supplement the initial information submitted in the CMC and other discipline to expand the scope of the investigation. What kind of information is submitted in information amendments? So all the changes in the drug substance and drug product process, or in the formulation of drug product, for example, that have the significant potential to affect the safety of the product, should be included and submitted to the agency in a timely manner in an information amendment. For example, when there are changes that might impact the impurity clearance or the viral safety, or there are changes in the source material and cell banks, or changes in the specifications, or in the container closure, or in the reference standards. Also, the holders of an IND, they need to report the progress of the investigation annually and 60 days, and they have to be submitted, this annual report, 60 days from the day of the IND, from the day the IND went into effect. And the annual report should include a summary of any significant manufacturing or microbiological changes that were made during the year and that were submitted in information amendments, and also should include updates of information that might not have been significant to submit an amendment, but they should be included in the annual report, like, for example, updates of stability. And this is basically a summary slide that to show the product life cycle. So sponsors during the research and development phase, they identify a candidate that they are going to manufacture a lot that probably is going to be used in tox studies. They do some limited characterization, some viral clearance, and limit stability, and use this lot in the tox studies. Safety information is collected, and then the sponsor will manufacture a lot that is going to be proposed for the clinical study for the phase 1. They submit the IND. The IND, if it is safe to proceed, then the clinical and the CMC development proceeds into phase 1, phase 2, phase 3. And then during this time, the sponsor collects more characterization data, they do assay development and validation, they improve and develop the manufacturing process, they do scale-ups, they tighten or collect more data to inform the release and stability specification, and they overall increase their understanding of the product and of the process. Basically, up to the point where there is sufficient information from all the disciplines to submit a BLA, the BLA is submitted. And this BLA package, we have extensive characterization studies, full process and assay validation, release and stability data, and final specifications. And so once approved, there is a lot of additional information for CMC because the sponsor might change places where the drug substance or drug products are manufactured, changes in formulations. And so there is a lot of activity that is also performed from a CMC perspective and also from a clinical perspective, potentially post-approval. And lastly, I want to just emphasize with this slide the importance of assessing the quality of the products. During development, the sponsors will manufacture several lots that they are going to be used in the clinical studies. These are the clinical lots. And basically, these lots, the sponsor will collect safety and efficacy information that will derive from these clinical lots used in the clinical studies. So the safety and the efficacy and the fact that the commercial lots have the same profile regarding safety and efficacy that the clinical lots is assured when there is a manufacturing process that is sufficiently and adequately controlled so as to be able to consistently deliver a lot of the expected quality. So if your manufacturing process is going to deliver every time that is run a product that has the same quality in the commercial space as far as in the clinical space, then the safety and efficacy information that you collected during your clinical studies can be leveraged to support that the commercial lots are going to have the same safety and efficacy profile that identified in the clinical studies. And this is key. So with that, well, of course, if you have any questions, I could answer them. And I really, really appreciate your time. This was long. I hope it was helpful. And please don't hesitate to contact me through my email address if you need any additional information. Thank you very much. I don't dare to pronounce it in Portuguese. That's why I have it written, but I will not say it.
Muito obrigada, Cecília. Foi muito wonderful presentation.
Thank you so much.
Eu tenho uma pergunta.
Sim, podemos?
O FDA tem alguma coisa escrita dizendo assim quais são as modificações que eles consideram substancial para se submeter à apreciação do FDA antes de ser implementado, ou só aquilo que ela mostrou ali, que são algumas partes, né, da regulação geral?
Cecilia, does FDA has any specific guideline for the sponsor to know the substantial amendments needed to be reviewed by FDA prior to implementation, or just the general information that you put in the presentation?
Thank you for the question. There is a guidance, and I have that linked, and it's about CMC information for phase 2 and 3 INDs. There is a link there. The other thing that I think I may clarify if it is not clear is the fact that the sponsor during an IND does not need to hear back from FDA to implement a change. From a CMC perspective, so they need. I mean, yes. So you make that's why it's very important that the amendments are submitted timely, and also it's very important that they are reviewed timely, because there is not a time frame there is not a clear timeline for coming back to a sponsor after an amendment. So it's very important that both stakeholders really do their job timely.
So for amendments, you are saying that there is no 30 days review, like a waiting period.
No.
Ok. Então, ela esclareceu que aqui no slide 79, que vocês vão receber a apresentação, tem um link para esse guia específico de INDs fase 2 em fase 3, CMC amendments, tá? E já tem o link aqui no rodapé do slide. Acho que fica claro. Ficou claro que não tem uma aprovação explícita do FDA, e também o FDA não tem o tempo dos 30 dias, não existe para emendas de CMC.
Não precisa da aprovação, né?
Não precisa da aprovação. Então, se o FDA achar que tem algum questionamento, alguma coisa, ele vai entrar em contato muito rápido com o sponsor. Cecília, just for us to know a little bit of like a time frame. So in case the sponsor submits a CMC amendment with a substantial modification, what would be like a regular feedback, like if FDA does not agree with that, how quickly would the FDA make some contacts with the sponsor?
Yes. So there are, in ODP, there were internal deadlines to review IND amendments. So the idea is that if you are the reviewer of an IND, you will receive notifications of the new amendments that are received. And as they are received, they should be looked and identified as if they are big amendments or like smaller amendments. And then based on that, the time frame for review. But this is an internal time frame.
Bom, primeiro eu vou agradecer a Cecília. Cecília, I would like to say thank you again for your time to prepare this presentation and be here today with us sharing your experience. And I can say from Ana Filgueiras and myself that it was a pleasure to work with you. And we really have learned a lot from you during this period. So thank you very much.
Hoje foi uma apresentação com muita profundidade, né? Acho que muito rica, muito importante, né? E aos poucos nós vamos aqui criando, aqui construindo, né, o nosso entendimento e sempre com o objetivo de tornar os nossos regulamentos mais claros, né, mais centrados na realidade, na nossa realidade. Eu acho que a Maria Cecilia trouxe informações muito importantes, né, inclusive as perguntas, enfim. Então, gostaria só de agradecer mais uma vez por isso. Continuamos, então, com esse trabalho que vocês têm feito, realmente muito importante, apesar de todas as circunstâncias, né? Nem sempre nós conseguimos que todos participem, né, aqui da COPEC, mas você já sabe, né, que com toda essa turbulência, mas todos seguramente vão assistir novamente, assistir depois, né, e caso tenham alguma dúvida, nós voltaremos com vocês. Vamos agendar a reunião, Camila e Beleza, para quinta-feira, para a gente discutir os outros pontos, né, das próximas etapas. E é isso. Muito obrigado mais uma vez. Maria Cecilia, thank you so much for your brilliant presentation. Muito obrigado a todos. Uma boa semana para nós.
Thank you so much.
Muito obrigada, gente. Foi uma participação muito boa da Anvisa. Agradecemos muito por vocês terem participado tão ativamente com as perguntas, que eu acho que é o que realmente traz o valor para esse momento que nós estamos aqui com o speaker e com essa disponibilidade para oferecer, e a gente também aprender. Eu acho que todo mundo das empresas da Interpharma que estão aqui, acho que conseguiram aprender muito sobre como realmente acontecem os processos no FDA. Então, foi muito um aprendizado para todos nós. Muito obrigada.
Obrigada.